How does anyone know where they finally belong? That's what I wanted to show, to whom I dedicate this song. It's a couple rarely seen. What were the chances that they met? It's a couple rarely seen. What were the chances they had met? Science or fiction? In between. No matter what, I had a bet. A and B fit well together. There is so much that they share. They were not created equal. Yes, their life was never fair. But they were there. We were aware. We took care, thus we had found them. More important, we could count them. They were hundreds, even thousands. Their special kind of relation, phosphorylation. Precondition for strong bonds and a sudden translocation. The equation can be solved, and we simulate their track. To answer an outstanding question, are they ever coming back? They were just two proteins regulating cell transcription, thereby writing their own story, following our prediction. Okay, so let's reconstruct what I just read to you. It's basically inspired by a paper from my master's, and there we investigated two transcription factors, STAT5A and B. That's why I'm asking, how does anyone know where they finally belong? And these two transcription factors, they form dimers, like pairs. So it's a couple, these two, but they are rarely seen. What are the chances they had met, I'm asking. And actually I started in this project with a probabilistic model calculating the chance or the probability that these two transcription factors form a pair or a dimer. So is it science or is it fiction? It's something in between. No matter what, I had a bet. So I was doing some calculations. A and B fit well together because if you look at their sequence, at their structure, they have homology around 80%. So there's so much that they share, structure-wise. However, they were not created equal, I'm saying, because they are not the same in cells. So you have much more A than you have B. So their life is not really fair. You cannot really compare them. But we were aware of this. We were there, we were aware. We took care and we found them, we detected them, we could measure them precisely, we could even count them. How many molecules are there? And we found hundreds, even thousands of these molecules. Now, in order to bind, in order for these transcription factors to form a couple pair, they need a special kind of relation, phosphorylation, which is the transfer of a chemical group to these proteins. And once they form a couple, a dimer, pair, they move from one part of the cell to another. And that's why I'm saying precondition for strong bonds, this phosphorylation, that they can form binding, and the sudden translocation, so that they move from one part of the cell to another. The equation, I did a deterministic model based on ordinary differential equations. The equation can be solved, even not analytically, but at least numerically, and we simulate their track. So we can really simulate on a computer how they move from one part of the cell to another one. And with this mathematical model, we can answer the question, are they ever coming back? So once they moved to the other part of the cell, are they ever coming back? And in this study we find they don't. They're never coming back. However, they were just two proteins, as I said, regulating cell transcription, so how the DNA is transcribed into mRNA. But transcription, of course, also literally means to write their own story. And actually their own story is following the prediction that I made, or the prediction that we made in this mathematical model. And that's how I got inspired by my first science.
Subscribe to the Science Talk to get your dose of science today.